And then let's welcome our next speaker, Stefan Berghoff. He was a research associate at the Max Planck Institute, joined the Klaus Army Naves lab to pursue his PhD studying cholesterol in the CNS. And he currently works as a postdoc in Mika Siemens lab in the German Center of Neurodegenerative Diseases at Munich. Stefan enjoys lo-fi and the trip-hop music. He formed a band called the Feiwa, where he plays guitar, compose songs together with his singer wife. And today he might not be playing music, but please join us to the Rock Zone to celebrate his science. You need to unmute.
No, thanks a lot. Sorry, couldn't unmute. Wait, I will quickly share my screen. The host has to allow me to share my screen.
Eugenio, can you help to that Stefan to share?
So here we go. Okay, now you should see everything. So thanks for this introduction. Thanks the community. Jerry, Lynn, thank you very much to inviting me to this really interesting session here. So the paper I'm going to present was published early this year, and it's entitled Microglia Facilitate Repair of Demyelinated Lesions via Post-Squalene Sterol Synthesis. I know it's not the most easiest titled, so I will try to go through that. And as you heard now a lot from Gary about the importance of buffering lipids and to form lipid droplets, I will somehow show you a mechanism how microglia loaded with cholesterol can remove these excess lipids. So I'd like to start with talking a little bit about cholesterol. And so outside the academic science world, I would say cholesterol is more depicted as being a really bad stuff. Which we all know as scientists is not the case. It's a really important and principal component of all plasma membranes, and it also serves as an intermediate for the synthesis of different important molecules. And when we have a look at the brain is actually a really cholesterol, actually the most cholesterol enriched organ. So it harbors around 1/4 of the whole body pool of cholesterol. And as we heard already, most of the cholesterol is located to myelin, which is a membrane formed by the oligodendrocyte and sheathing the axon and allowing nerve conduction. So with regards to cholesterol metabolism inside the CNS, it involves many different cell types. And I just want to highlight 4 important, let's say, facts about the cholesterol metabolism in the CNS. So, and first of all, cholesterol has to be synthesized locally. And this is due to the specific properties of the blood-brain barrier. This restricts the entry of peripheral cholesterol into the CNS. And it is still considered that astrocytes are the main producers of cholesterol and transfer it via HDL-like particles to other cell types. For instance, here depicted the oligodendrocyte, for instance, for myelin membrane synthesis. As Gary already told you, cholesterol can't be eliminated, so it has to be converted either to a more hydrophilic version to leave the CNS. So this is these hydrophilic versions are called oxysterols, and it's considered that most of this conversion is done in neurons. And there might be also an alternative route that involves ApoE. So cholesterol synthesis is not the most easiest metabolic pathway. It involves over, I think, 27 different enzymes, and it's also an energy demanding process. And I will not go through this whole process now. I just will reduce a little bit the complexity here and highlight 4 important enzymes of the pathway that are also heavily regulated. And starting with the first important one, which is HNG-CoA reductase, it's also the rate limiting step of the pathway. And most of the people know this enzyme due to a class of compounds, inhibitors, which are called statins, that have given us the opportunity to treat hypercholesterolemia. The second important enzyme is squalene synthase. So it catalyzes the first reaction that is committed to cholesterol. Then the cholesterol synthesis pathway branches in two different sites. One is called the Bloch pathway. So this is mediated via llanosterol 14-alpha demethylase, leading to a number of intermediates with an unsaturated side chain. And then another important enzyme is 24-dehydrocholesterol reductase, which leads to several intermediates with a saturated side chain. And it's also the terminal cholesterol synthesis enzyme converting desmosterol to cholesterol. So from now on, I will just show this simplified version of the cholesterol synthesis pathway depicting those four important enzymes. So as Gary already introduced to us, a really severe disease of the CNS, which is called multiple sclerosis, which leads to the destruction of the lipid-rich myelin sheet as depicted here. So this involves immune cells, but also macrophages that are also involved in clearing the myelin debris. So, and what we want to know in our study is, following a demyelinating episode, how cholesterol metabolism is altered, and even more important, how is the contribution of the cell type specific cholesterol synthesis to remyelination. So, and to answer this question, first of all, I thought it might be useful to introduce quickly the animal models of multiple sclerosis that we have in the lab, because there are plenty of one, and I will just now split them into the toxin-induced demyelination models and the inflammatory demyelination models. So on the toxin-induced demyelination models, that gave us strong tools, especially to focus on the remyelination situation. We have one, the cuprizone mouse model, and the lysolecithin focal lesion model that has been already shown in the previous talks. And for the inflammatory models, we have one particular important one is the experimental autoimmune encephalitis, so EAE, which is achieved by immunizing the animal against myelin antigens. And I would just quickly go a little bit more in detail about the cuprizone model, because it also has a great advantage of having two different episodes of de and remyelination. So after a certain while of giving this toxin to the animal, here around 5 weeks, we end up with a nearly complete demyelination of the corpus callosum, and then spontaneous remyelination occurs, peaking at around 6 weeks. If the cuprizone diet is continued, again, demyelination will take place, and there will be a small peak of remyelination at around 12 weeks. And to at least achieve a partial remyelination of the corpus callosum, we remove cuprizone here from the diet. And you see it here, after 2 weeks, we end up with at least a partial remyelination. So with this model, we have the opportunity to analyze two different phases of de and remyelination. And with this, we wanted to get a better impression what happens if a lesion exhausts. So we experimentally, we did a lot of expression profiling of tissue, but also isolated glia cells of cuprizone demyelinated animals at several time points. We created several cholesterol synthesis mutants by targeting squalene synthase as the first committed step to sterol synthesis. And we also manipulated the sterol availability by either using an intermediate or by using cholesterol. And I already will summarize here a major finding that we had. And this was somehow also a surprising finding, that there's such a huge difference between an acute remyelination phase and a chronic remyelination phase. So during the acute remyelination phase, the functional pathway of sterol synthesis in microglia seemed to be really important. while in contrast, in the chronic phase, oligodendroglial sterol synthesis seems to be the main driver of remyelination. So the finding in the chronic phase was maybe not that surprising, since in our previous story from 2017, we already saw that by increasing cholesterol availability, this was specifically helpful during the chronic disease phase. And also by another group, it has been shown that after chronic demyelination, cholesterol synthesis is upregulated in oligodendrocytes. But what really puzzled us was the effect in the acute remyelination phase. So just to illustrate what we actually saw is that following genetic elimination of squalene synthase in microglia, we ended up with a picture of failed remyelination, which is depicted here by Goyard-Schmalin silver staining. And we had a massive ongoing microgliosis. When we looked at semithin sections and also via electron microscopy, we saw a lot of foam cells appearing and also increased number of crystals. This also, when we isolated the first tissue samples and did a rough qPCR profiling, we saw that the main cholesterol efflux transporters, ABCA1 and G1, were downregulated in comparison to controls. And we found an increased pro-inflammatory signature, like for instance here for NOS2, and also reduction of anti-inflammatory genes such as interleukin 10. So in the end, we ended up with, following the knockout, we ended up with a picture that we had a reduction of cholesterol export and an increased inflammatory response. So at that time, this puzzled us a lot, and we really were going through literature, testing different hypotheses. And then there was one interesting observation that brought us on the right track. So already in 2012, in a group around Christopher Klaas, they have identified in macrophages of arteriosclerotic lesions an interesting pathway that actually links cholesterol export, inflammation, and also the functionality of the sterol synthesis pathway. So, and the important signaling axis here are the liver X receptors. So what they have shown is that following cholesterol loading of a macrophage, there is accumulation of a cholesterol intermediate called desmosterol. And this intermediate has a strong LXR ligand function and allows the increased expression of cholesterol efflux genes and also limits inflammation. So according to their finding, when we knock out sterol synthesis at a level of squalene synthase, it could be that we lose the synthesis of the LXR ligand desmosterol and end up with the picture that we see here, a foam cell that is pro-inflammatory. So we tested this and isolated microglia from cuprizone demyelinated animals and checked some cholesterol synthesis genes. And what we saw is a drastic reduction in the expression of the sterol synthesis enzyme DHCR24, so depicted here. We then measured sterol abundance in the tissue of those animals and indeed found increased abundance of desmosterol, while, for instance, latosterol that should not be formed under this situation was reduced. To verify our finding, we again isolated microglia and measured sterols within those cells. And again, here we found increased abundance of the LXR ligand desmosterol. Proving that we were on the right track in our cholesterol sterol synthesis mutants, we found we didn't find anymore the increase of desmosterol within tissue. And also we found a reduction of the expression of LXR target genes such as ABCA1. So to prove that this is not a specific effect in the cuprizone model, we also analyzed samples from EAE, lysolecithin, but also a genetic demyelination model, and indeed found evidence of this pathway in all these different demyelinating models. So then we wanted to exploit how we can use this pathway in terms of a therapeutic option. And in our case, we were thinking about an intermediate, which is squalene, as a natural occurring lipid that might actually feed into desmosterol synthesis and induce LXR activation. And we first of all tested this in the EAE model. So these animals were treated after the onset of disease, peripherally with squalene in the diet. And indeed, we saw a strong reduction of the disease expression. So this translated also in reduced lesion volume, here labeled by IBA1 in light sheet microscopy. And when we isolated microglia from those animals, we indeed found upregulation of LXR target genes and downregulation of inflammatory genes. So squalene has another benefit, because it might not only be reducing the pathology in terms of targeting the microglia macrophages, but it might also support oligodendrocytes, since it can be metabolized to cholesterol and thereby used for myelin membrane synthesis. So we tested this first of all in an in vitro system, spinal cord co-culture model. So these cultures were treated with squalene, and then the appearance of MVP positive myelin sheets was counted, and indeed, as you see here, we found supported myelination in these cultures. And also in the lysolecithin, when we quantified the appearance of mature oligodendrocytes labeled with CA2 within the lesion area, we find increased numbers of oligodendrocytes. So supporting the idea that squalene might actually, in addition to target microglia macrophages, support oligodendrocytes as a substrate. So we also tested this therapeutic approach in a combination therapy idea, and combined squalene therapy with interferon 1 beta as the classic first-line treatment. And indeed, we saw a strong reduction of the disease severity by combination therapy. And as a proof of principle, we also combined the squalene treatment with a desmosterol mimetic. And as you see here, we were able to achieve increased LXR targeting. So the final question we had is, can we translate our finding to the human pathology? And for that, we took advantage of public available data sets, so single cell data sets, here by Sarah Jäkel and also Masuda. And our bioinformatician expert Ting reanalyzed these two data sets with the focus on microglia macrophages. And following the integration and the identification of those 4 clusters, we identified this cluster 2 here that we termed lipid export clusters, because in the MS samples, we found here a strong reduction of DHCR24 as the terminal cholesterol synthesis gene, and also the increased expression of LXR target genes such as ABCA1 and ApoE, as well as LXR alpha and beta. So that within the MS lesion, there's active LXR signaling is not completely novel. So it has been nicely shown here by co-labeling or by labeling for LXR alpha already 2017, and also by labeling for ABCA1, that indeed there's the active LXR signaling within the MS lesions. And more interestingly, by comparing the expression profile of a chronic active lesion rim versus an inactive lesion rim, DHCR24 downregulation is one of the top candidates. So this prompted us to get hands-on MS tissue and isolate different lesion areas, so the lesion rim and the lesion center, of active and inactive lesions determined by CD68 labeling. So as you see here, regardless of the lesion type, the abundance of cholesterol is drastically reduced. In contrast, when we checked the active lesion rim and the active lesion center, we found increased abundance of the LXR ligand desmosterol, while in the inactive lesion center, this intermediate was reduced. And irregardless of the lesion type, latosterol, for instance, was decreased. So I hope with this data, I can convince you that synthesis of the sterol precursor and LXR ligand desmosterol is a conserved pathway in phagocytes following myelin uptake, that squalene supplementation feeds into desmosterol synthesis, and that desmosterol synthesis can be found in phagocytes of human MS lesions. and also that squalene might be an interesting way for a combination therapy approach in MS patients. So with this, I'd like to thank my former mentors, Klaus Avinave and Gesine Saher. The whole group of young PhD students that was involved in this project, especially Lena Speed, who was involved in nearly all of the experiment. I'd like to thank the collaborators, especially Julia Edgar for her work in the, on the myelinating co-culture model. My new boss, and at that time former collaborator, Michael Simons, and also Ludovico, and Christine Stadelmann and Franziska Papst, who were involved in isolating the MS lesions. So with this, I'd like to thank you. And I also wanted to highlight an upcoming next story, which closes or which focuses now on the last cell type that we so far missed, which are the neurons. So thanks a lot.
Cool. Thank you, Stefan. This is very like a complicated pathway.
I know. It's not the most easiest one, yeah.
Yeah, my PhD also did a little bit of work with the cholesterol. It's like, yeah, when I look into the literature, it's like, wow, it's overwhelming, and it did a fantastic job to like tease this apart. Yeah, actually, I have a question. Like in the early slides, you mentioned that early repair, remyelination, it's more of like a microglia side of like a cholesterol, like digestion, and then the chronic one, it's at the oligodendrocytes of the pathway. I'm curious, does that is meaning it's more of like integration part of like to generate the myelin, or you think that it's the pool of that population of remyelinated oligodendrocytes that have it in the plays a main role in the cholesterol synthesis pathways coming from like the differentiating oligodendrocyte lineage in general, or like a survival oligodendrocyte, they also do something to actively repairing their myelin sheath.
So at the moment, it looks like that in an acute lesion where we have also this massive microglial response, most of the cholesterol, but also other lipids, is recycled. So that there's really no need to synthesize cholesterol because there is so much cholesterol. So it's just recycled by the microglia, and this is really done efficiently. So also when we compare this acute and chronic phase in terms of efficiency with time, it just takes 1 week to remyelinate a lot of, and in the later time points it takes a long time, which also hints to direction. So if we look at developmental myelination, this is a long process, and their oligodendrocyte sterol synthesis is really important. So that's why I think in the chronic situation where, first of all, the microglia might be also exhausted, oligodendrocyte need to start the old developmental process for making up myelin again. So this is at the moment how I see it, at least.
So cool. And then I missed this. Then the squalene treatment to the EAE animal. How do you treat it in a chow or injection, something like that?
So the advantage of squalene, it's an oil. And you can just add it to the food. So it's really the easiest treatment you can do. Yeah, so I think that's already answering the question. Yeah, so, and we tried different paradigms like prophylactic, therapeutic, and so on, and it always shows a really robust reduction of the disease expression, and especially in the EAE model.
I guess you get this question a lot, like what type of human food that have a lot of squalene will you recommend us to take some of those, like just in case?
So I'm a fan of good Greek olive oil. So this contains a lot of squalene, and actually, so this is just a really interesting information that I got by going through some papers. If you compare the mean ratio of squalene intake between America and, for instance, the Mediterranean countries, there's a difference of around, I think in USA it was around 40 milligram or something, and the Mediterranean countries something like 400. So this is really the difference there. And I know this is maybe a little bit too far, but we all know how good the Mediterranean diet works in different pathologies, and maybe squalene is one part of that story.
Right. Yeah. Next step is to compare like two countries, like population of in terms of the neuroinflammation disease, and see if that is significant association. Right? Yeah. Is there any questions from our audience? You can unmute yourself and start the video to interact with the speaker. Oh, yeah, we are almost time. That's really a great talk.
Thanks a lot. Yeah, I think I was a little bit over the time, so people are leaving. It's late, at least here.
Probably they're hungry.
Yeah, or they're hungry.
Eugenia?
Yes, well, thank you very much for your participation today. In the name of Jerry and Open Book Science, I thank you all of them. Thank you, our great host today, Jing-Ping Lin, and all the great talks today. And yes, follow our social networks and hopefully see you soon next time. Thank you.
Thank you.
Bye.
Thank you. Looking forward to your follow-up stories. And yeah.
Thank you.
Bye.
You too.
Bye.
Ciao, ciao.